All right, let's jump straight into this explainer. Imagine for a second we're standing out in the hallway, right in the middle of morning teaching rounds. We've just stepped out of the room of a patient who's presenting with early acute kidney injury, AKI, right after a pretty severe episode of ischemic shock. Now their blood pressure is finally stable, but their kidney function, it's rapidly declining. To understand exactly why that's happening, we can't just look at the big picture. We need to zoom in, way in. We're going to leave macro hemodynamics at the door today and dive deep into the microenvironment of the kidney itself. We're looking at the cellular and molecular injury phase of AKI, and honestly, it is a fascinating cascade. We're talking structural collapse, microscopic traffic jams, and literal explosive cell death. So grab your metaphorical clipboards and let's get into it.
Okay, let's kick things off with our first interactive rounds question. Doctor, I want you to picture the proximal tubular epithelial cell. This thing is highly active. It is absolutely packed with mitochondria, and it relies completely on oxygen. Now, during the initial sublethal phase of ischemic injury, this cell goes through severe ATP depletion. It's an energy crisis. So my question for you is, physically, on a molecular level, what actually happens to the sodium-potassium ATPase pumps during this crisis? Take a second to think about the structural integrity of the cell itself.
Well, here's the answer, and it's wild because it's a purely mechanical failure. When ATP tanks, the actin scaffolding inside the cell literally starts to dismantle itself. It depolymerizes, and this is driven by a protein called ADF or cofilin. Basically, the cell's internal framework just collapses. Normally, your sodium-potassium pumps are anchored really tightly to the basolateral membrane, the blood side of the cell. But without that actin scaffolding, they detach. The cell entirely loses its polarity. These pumps physically drift and mislocalize all the way to the apical membrane, the side facing the tubular lumen. And because they're stuck on the wrong side now, they drive bidirectional transport, meaning they're pumping sodium right back out into the urine.
Now, why does this matter for us at the bedside? Because this microscopic misdirection has an immediate, measurable clinical consequence. When you order labs on our patient and calculate their fractional excretion of sodium, their FeNA, it's going to come back high, usually over 1%. That high FeNA isn't just a random lab abnormality. It is the direct, physical result of those mislocalized pumps literally dumping sodium back into the lumen. And to make matters worse, the cell is still desperately burning through its remaining precious ATP to do this. So it's wasting energy right when it needs it the absolute most. It's just a vicious cycle.
So as we zoom out just a tiny bit from that epithelial lining to the microvasculature right next door, we enter what's called the extension phase of AKI. And this is where endothelial cell injury and dysfunction takes center stage. You see, it's not just the tubular cells that are suffering here. The blood vessels themselves are fundamentally damaged by the ischemic event, and they actively extend that zone of injury.
Which brings us to our second big question for rounds today. Our patient had a hypotensive episode, right? But the ICU team gave them fluids, they used pressors, and now their systemic blood pressure is totally back to normal. Yet, if you look at the kidney tissue, specifically the outer medulla, it is still severely hypoxic. We call this the no reflow phenomenon. So how do you explain this? Why hasn't the microscopic blood flow returned even though the big picture macro hemodynamics are totally fixed?
The answer is that the microvasculature is essentially gridlocked. Even with normal blood pressure, those tiny endothelial cells lining the vessels have been injured and they physically swell up. They shed their glycocalyx. Red blood cells get trapped in there, clumping together in a stack called rouleau formation. On top of that, white blood cells, leukocytes, start sticking to the vessel walls and plugging up the vasa recta. It's a massive microvascular traffic jam. And the S3 segment of the proximal tubule, which sits right in the outer medulla, gets hit the absolute hardest by this. It's starving for oxygen, but it's trapped in this congested low-flow capillary bed long after the patient's systemic blood pressure recovers.
Eventually, if this ischemia and microvascular congestion drag on, the cell hits a point of no return. What started as sublethal injury becomes lethal. But before it dies, the cell tries to protect itself. It attempts to halt the cell cycle entirely to avoid replicating any damaged DNA. So, doctor, if you were evaluating this patient with early AKI, what are the two most validated urinary biomarkers we can look for to catch this specific cell cycle arrest? That's right, TIMP2 and IGFB7. Spotting those tells us severe AKI is progressing. Now, if the damage blows right past that arrest phase and becomes too severe, the cell is forced to die, and it has to choose between two vastly different paths, apoptosis or regulated necrosis. Time for our third interactive challenge. Let's say we put these dying cells under an electron microscope, and we analyze their chemical signaling. How would you distinguish between apoptosis and a specific, highly regulated form of necrosis called necroptosis? What are the core mechanistic and morphological differences between a cell undergoing apoptosis versus one going through necroptosis?
The contrast here is honestly mind-blowing. Apoptosis is the neat, tidy, quiet way to die. It actually requires energy to execute perfectly. The cell condenses its nucleus, it shrinks down, it forms these little membrane-bound blebs, but crucially, the plasma membrane stays completely intact. The cell just neatly packages itself up to be consumed. Necroptosis, on the other hand, is violent. It's a genetically regulated pathway driven by a kinase called RIPK3, and it leads to massive swelling of organelles, vacuolization, and, the real kicker, the explosive rupture of the plasma membrane.
Now, that explosive membrane rupture in necroptosis isn't just a structural failure. It is a profound immunological alarm bell, leading us to the concept of necroinflammation. When that cell bursts open, it dumps all of its raw, unprocessed intracellular contents straight into the kidney interstitium. We're talking organelles, DNA, RNA, and highly immunogenic proteins. We call this spilled material damage-associated molecular patterns, or DAMPs. They act like molecular shrapnel, setting off massive inflammatory alarms all throughout the surrounding kidney tissue.
This whole process aligns perfectly with a brilliant conceptual framework proposed by Polly Matzinger called the danger model. Traditionally, we're taught that our immune system only attacks foreign invaders, right? Bacteria, viruses, things like that. But the danger model argues that our innate immune system is heavily hardwired to respond to our own molecules if they end up in the wrong place. So when DAMPs are released from dying cells during an ischemic event, which is a sterile injury, no bacteria involved, the immune system reads that molecular shrapnel as extreme danger and launches an aggressive attack anyway.
So you might be wondering who exactly is listening for these danger signals. It's the mononuclear phagocytic system. Basically, dendritic cells and macrophages form this dense, interconnected sensing network that sits right in the interstitial space, wedged exactly between the tubular epithelial cells and the peritubular blood vessels. Think of them as the kidney's armed security guards. Because they are right there on the front lines, they have instant access to any DAMPs released by ruptured cells. They sense the damage, they sound the alarm, and they start recruiting even more inflammatory cells into the kidney.
But here is the catch. These security guards have dual personalities. Macrophages in this microenvironment can polarize into two wildly different states. You have your M1 macrophages, which are the classic pro-inflammatory attackers. When danger signals activate them, they pump out aggressive cytokines like TNF-alpha and IL-6, which literally drives further tissue injury. But as the timeline of AKI progresses, we hopefully see a shift toward M2 macrophages. These are the alternative, anti-inflammatory healers. They're activated differently, and they produce growth factors like IGF-1 to help resolve the inflammation and start repairing the tissue. So the balance between M1 and M2 is literally the balance between extending the kidney injury and finally starting the healing process.
Which leaves us with one final, pretty provocative thought to wrap up our rounds today. We've seen how cells dying via necroptosis rupture and spew those highly immunogenic DAMPs, causing massive damaging necroinflammation. We've also seen how apoptosis neatly packages everything up safely. So if we know that severe injury forces a cell to die no matter what, how might future therapies intercept that decision? Imagine if we could use targeted drugs to block RIPK3, forcing a dying tubular cell away from that explosive necroptosis pathway and steering it into quiet apoptosis instead. Could shifting a deadly signal from a highly inflammatory pathway to a silent one be the ultimate key to stopping the devastating extension phase of AKI? It is an absolutely thrilling frontier in nephrology. Thank you so much for joining me on Rounds today. I really hope this explainer has totally transformed how you visualize the molecular battlefield of acute kidney injury. Keep asking those great questions, and I'll see you next time.